Thank you, everyone. My name is Rafael Vescovi. I'm going to present today our discoveries on working on a national laboratory for the US government and going from a single instrument to a science facility. So a little bit of background, like my presentation is going to be very different, and hopefully like this will be the naive view of like how to automate a laboratory. Where's the presentation here? Sorry. Say it again.
It's at the bottom right.
Yeah, but I can.
Yeah.
So, on autonomous discovery, like we usually like to think as two loops. So you have the inner loop where you have like synthesis, the experiments, the measurements, everything that a scientist do inside a laboratory until it acquires data. And then we have the outer loop, which is everything that we do like with creative thinking. Like is the models that we use, is the insight that the PI have of saying like, that's going to be my next sample or that's going to be my next step. And we want to bring these two together. So we have like very different names. Some people are calling it smart autonomous integrated laboratories, self-driving laboratories, autonomous discovery. But at the end of the day, all we want is to have a laboratory that we can use as a program and that we can change like a recipe repetitively and do like some kind of repetitive science to go forward. So self-driving laboratories needs very, very different capabilities. Like we need like the experiment model on one side. We need to create samples, we need to synthesize, and we need to do measurement. And those facilities like may be very different, especially compared to the compute facility. So at Argonne National Laboratory, we have the largest AI computer in the world. And part of like how this project started was, well, we have all these good computer scientists. Can we bring other laboratories that already existed, that already got all the money invested on it into the automated, automatic, autonomous world? And we found that those were very, very different researchers with very, very different languages. So we started the Argonne Autonomous Research Laboratories, AROS, and we have the Airfree, which is inside a glove box. We have the Rapid Prototyping Laboratory, which is a showroom for automation that cannot use any chemical. We have like the APS with X-ray sciences, like in this gigantic accelerator. And we have a biosafety level 2 facility. So those are completely different facilities, different scientific instruments, and completely different safety like requirements. And bringing those together was like the challenge that we have. So one of like our initial examples is to generate peptides using AI, and then see which of these peptides like can kill bacteria from biosafety level 1 and biosafety level 2, like antibiotic resistant bacteria, see if like how these peptides react, update the model, synthesize and continue this experiment. So this was like the motivator experiment behind this. But that's not what I'm going to talk about. I want to talk about a much, much simpler experiment. I want to mix printer ink. And mixing printer ink sounds like a very simple, you go on your printer, you put in the RGB code, and it brings out any color that you want. So we try to make that as complicated as possible. So we built a whole laboratory to mix printer ink, and it gets a plate, tries to mix new colors, updates a genetic algorithm, and keeps doing that until it finds the color that you asked. So what was the first finding? The first finding is there is an application that we defined in Python that have very, very large verbs, which is mix colors, or run an experiment, or analyze a plate. And then we have like this middle layer, which is like what's interacting with the instruments, with the robots. And these like are easy to describe linear sequence of actions, and then suddenly these actions go into the instruments itself, and each of these instruments is completely different. We have like very nice open source like mixers. We have like a USB camera that is very easy to use, and then we have a Windows Communication Foundation from 1990 that never had the software updated, that all needs to communicate together. So this is the outer loop, right? Like this is the steering, the reasoning, like the discovery is all on the Python side. The inner loop, like the common actions, like how we define our workflows, like how we're going to schedule things, like do the data processing. That's part of the inner loop, and we found that there's one more person that needs to work. So is the component specific? Like, is the guy that is going to, like, the specialist on the instrument, on the robot, that is going to make like this robot do that action very, very well. So I came from science like 15 years ago. My laboratory had one person that was these three. Like it was one computer and one person that knew how to do everything, and if like this person was on vacation and something went wrong, everything stopped. We want to separate that as much as possible. How do we separate that? Well, that's not so easy, because like now we have like these data people, we have like this robotics, like these instrumentation people, and we have a biolab. Not every biolab is going to have the budget to hire all these scientists, and we don't have the training or like even like the capacity to provide all these people, right? So that came to our paper, like towards a modular architecture for science facilities, and it's a lot of like repetition, like of what a lot of other laboratories already did, but with like the learnings that we had on trying to train these people, on trying to get this to work. So the first thing is like we said like, let's build this gigantic like software infrastructure that takes care of everything. And we are going to have configurations for the work cell, for the workflows. We are going to use human readable, like we're going to use AI. And we are going to have instruments that can use epics, REST, TCP, ROS1, ROS2. And the first thing that we found was that we don't want that. Like, that is way too complicated. That involves a lot of people having, that involves a lot of engineering that we didn't have the capacity to do. So we went like one step further and said like, how can we use what's already on the market? Like we have Blue Sky, we have like the robotic chemists, like we have a lot of laboratories that already have like good scheduling system, that already have drivers. How can we integrate all that into our work? So we decided to future-proof and try something much simpler. We're just gonna enforce how we connect to our instruments. And this is a little bit like of the minimal way that we want to interact with our instruments. First, every single instrument in the laboratory needs to have an endpoint that tells what they are. So I'm a chem speed and I can do liquid mixing. This is the comments that I receive. Like, second, it always needs to have an endpoint for an action. And this action needs to be the action that was broadcasted. Like a sealer can only seal. And this needs to be very, very well defined. The second is, the third one is it needs to tell me I am busy, I'm ready, or I have an error. Nothing more complicated than that. And fourth, like, which is a little bit more complicated, it needs to tell me the resources. And especially for DEC robots, like ChemSpeeds or like the Unchained Big Kahuna or the OT2s, I need to know what's on the deck. Otherwise, like, that's gonna be very complicated. Because I'm a data scientist, I decided to try like to put that into FAIR principles. So FAIR means findable, accessible, interoperable, and reusable. And this is how we define data for data science. Like, it's usually very hard to have a data set that is fully fair. But I think like we have enough engineering power in this room and in the many conferences that I've been to make at least like the implementation of instruments for autonomous discovery as fair as possible. So what is findable for me? Findable for me means like every instrument have a very well-defined address, accessible, like instruments can be used by any scheduler as long as like they are available. Interoperable means that it doesn't really matter what you're using as your scheduler as long as you know how to like the REST messages goes. And reusable means for me is if I have a new instrument and I need to write a template for it, I want to control copy it and change as little lines of code as possible to get this new instrument into my laboratory. So we decided to create like our layered infrastructure. This is like a little bit of how it will look like. We have a REST template for pretty much everything that is Python based. And all you need to do on this REST template is change the about, change the actions, and change the resources, change the state, and connect it downstream to whatever is on your computer. Now, we thought that this was going to be the jack of all trades and solve it, like all the problems. We also had to create a C# like a template because a lot of like the bio instruments, a lot of like the instruments that we are using comes with Windows DLLs. And there is a monetary equation here, which is having an engineer work 6 months like to integrate an equipment, it's very expensive. So if we already have like these two templates and we can minimize this engineering time, that can minimize like the amount of money that allows Lean to invest to become automated. So we actually used a lot of robots. Like we have Brooks BF400s, UR16.5, XPills, Sealers, ChemSpeed, Unchained Big Kahuna, OT2s, OT-Flex, Hudson Robotics, like a lot of like these like are complicated to integrate, a lot are very easy. What we created is just a repository that everything that we did is open source and is pretty well described. Now, before you guys go on the repositories and say like, oh, there's code from 15 generations, that's true. Like, unfortunately, it's very hard for us to keep all this code like up to date, and sometimes like we have like students working or like programmers. So we want to at least make some effort to have like everyone that is using the same instruments contributing to the same place. And that doesn't need to be us, but someone needs to do it. So so we created a workflow abstraction system, and a lot like it was shown before, our workflow abstraction was designed to be completely human readable and completely interoperable with other schedulers. And because I'm a computer scientist, I like the idea that every function has a target, or let's call a name, a target, and a set of variables. And that's how my workflows like are pedantically written and complete like in terms of science. So what we ended up showing is that creates some kind of like ontology between laboratories, and we can get the same workflow and move to a different equipment, move to a different laboratory, and all I need to do is change the targets. Like, I don't care if it's a PF400 or a UR5 doing a transfer. A transfer sample is a pick and place that happens from position 1 to position B, and for the scientists, like, that shouldn't matter. So this is what like we are trying to create, like to abstract out of like this language. So some of the examples here, like we have like our showroom laboratory. Like this showroom laboratory is doing the color picker. The first thing that it did was it grabbed a plate, it takes a picture, like it checks if the plate is empty, it moves into the OT2, try to mix some colors, moves this plate back, and then here there's something funny that happens. This plate like takes a JPEG image and sends that to the largest AI supercomputer in the world and allocate a nodes on that computer to analyze that plate. Takes more time to send the plate than to run the code, and sends it back. Like, why is this experiment so important? because like cross-facility data transfer and cross-facility policies, especially with DOE facilities, is very complicated. So we are showing here that you can do all that without the user or without the scientists have to worry about what's happening. And of course, like everything gets published to the user in a crunchable way, and the user only like looks at the graph like that's happening on the end. We've been using Gazebo and NVIDIA Omniverse, and one of the biggest things that we found is like if we're going to have like this gigantic science factory, like we need to test like how things will look in space, like how the movements are going to go. We need to train things to go a little bit forward. So how this science factory will look like, and this is the dream that we have, is a user will come and ask for a scientific recipe. Like we are gonna interpret this recipe, multiplex it, create actions that are independent from the robots, and then have like a gigantic floor room that starts to generate the data, it starts to reconfigure itself, and only provides to the end user like the full set of of data for the end experiment. So here is also another example of what we are doing. This is an X-ray environment like the NSLS2 that Phil showed, and we are using like these UR robots to build a sample, actually pipette this sample and put on a beam line, process all the data, send to the supercomputer, and get it back without any user, without any human interaction. Now, why is this so important? If you guys use it any kind of like X-ray or nuclear facility, it takes more time to close the door than it takes to get the measurement. And with the updates, like the government spending billions of dollars to update these facilities, data acquisition became between 10,000 and 100,000 times faster. So this opening the door, closing the door now became like a huge, huge time sink for these facilities. So having like robots that can interact with the beam lines is like a big part of the future that we have. This is not the work of a single person. Again, as I said, I started this as a naive contributor to trying to automate all these laboratories, but there's this huge team of people funded by the DOE, DOD, and other sources. And thank you.